Transport in plants, Part 2. You might have carried out an experiment at some earlier stage where you had placed a twig bearing white flowers in colored water and had watched it turn color. The cut end of the twig, on examining, you had noted after a few hours the region through which the colored water moved. Very easily, that experiment demonstrates that the path of water movement, more specifically, is through the vascular bundles, the xylem. Now we have to try and understand the mechanism of movement of water and other substances up a plant. Within a plant, long-distance transport of substances cannot be by diffusion alone. Diffusion is a slow process. It can account for movement of molecules only for a short distance. For example, it takes approximately 2.5 seconds for the movement of a molecule across a typical plant cell. At this rate, can you calculate how many years would it take within a plant over a distance of 1 meter for the movement of molecules by diffusion alone? Often substances have to be moved across very large distances in large and complex organisms. Sometimes diffusion or active transport would not suffice as the sites of production or absorption and sites of storage are too far from each other, so as to move substances across long distances, and at a much faster rate special long distance transport system become necessary. Generally, by a mass or bulk flow system, water, minerals, and food are moved. Mass flow is the movement of substances in bulk or en masse from one point to another between the 2 points as a result of pressure differences. Substances, whether in solution or in suspension, are swept along at the same pace as in a flowing river, which is a characteristic of mass flow. Different substances move independently depending on their concentration gradients, which is unlike diffusion. Either through a positive hydrostatic pressure gradient or a negative hydrostatic pressure gradient, the bulk flow can be achieved. The bulk movement of substances through the conducting or vascular tissues of plants is called translocation. Highly specialized vascular tissues are present in higher plants, xylem and phloem. From roots to the aerial parts of the plant, xylem is mainly associated with translocation of water, mineral salts, some organic nitrogen and hormones. A variety of organic and inorganic solutes are translocated by the phloem mainly from the leaves to other parts of the plants. How do plants absorb water? We know that most of the water that goes into plants is absorbed through the roots. That is why obviously we do not apply water on the leaves but apply water to the soil. Specifically, the root hairs that are present in millions at the tips of the roots are responsible for the absorption of water and minerals. The surface area for absorption is greatly increased by the root hairs, which are thin-walled, slender extensions of root epidermal cells. The root hairs absorb water along with mineral solutes purely by diffusion. Once the root hairs absorb water by two distinct pathways, it can move deeper into root layers. Apoplast pathway, symplast pathway. The system of adjacent cell walls is the apoplast that is continuous throughout the plant except at the Casparian strips of the endodermis in the roots. The apoplastic movement of water occurs exclusively through the walls of the cells and the intercellular spaces. Movement through the apoplast does not involve crossing the cell membrane. On the gradient depends this movement. Water movement is through mass flow and the apoplast does not provide any barrier to water movement. The intercellular spaces or the atmosphere into which the water evaporates, tension develops in the continuous stream of water in the apoplast. Hence, due to the adhesive and cohesive properties of water, mass flow of water occurs. The system of interconnected protoplasts is the symplastic system. The water travels through the cells during symplastic movement, their cytoplasm. Intercellular movement is through the plasmodesmata. The movement is relatively slower as the water has to enter the cells through the cell membrane. Movement is again down a potential gradient. Aided by cytoplasmic streaming is the symplastic movement. The cytoplasmic streaming may be observed in cells of a hydrilla leaf. Due to streaming, the movement of chloroplast is easily visible. In the roots, most of the water flow occurs via the apoplast as the cortical cells are loosely packed, and hence to the water movement no resistance is offered. The inner boundary of the cortex and the endodermis, however, is impervious to water because of the Casparian strips, a band of suberized matrix. As the water molecules cannot penetrate the layer, they are directed to wall regions which are not suberized into the cell proper through the membranes. Through the symplast, the water moves and crosses the membrane again to reach the xylem cells. In the endodermis, the movement of water through the root layers is ultimately symplastic. Water and other solutes can enter the vascular cylinder only in this way. Water is free again once inside the xylem to move between cells as well as through them. Water enters directly into the xylem vessels and or tracheids in young roots. These are non-living conduits and so are parts of the apoplast. Some plants have associated with them some additional structures that help in water and mineral absorption. A symbiotic association of a fungus with a root system is a mycorrhiza. Around the young root, the fungal filaments form a network or they penetrate the root cells. The hyphae have a very large surface area. From a much larger volume of soil, the hyphae absorb water and mineral ions that perhaps a root cannot do. The minerals and water are provided by the fungus to the roots. In turn, sugars and nitrogen containing compounds are provided by the roots to the mycorrhizae. Some plants with the mycorrhizae have an obligate association. For example, without the presence of mycorrhizae, pinus seeds cannot germinate and establish. Water movement up a plant. We looked at from the soil how plants absorb water and move it into the vascular tissues. We now have to try and understand various parts of the plant, how this water is transported. Is the water movement still passive or is it active? What provides the energy for the water to be moved up a stem against gravity? Root pressure into the vascular tissues of the roots. As various ions from the soil are actively transported, water follows and increases the pressure inside the xylem. This positive pressure is called root pressure, and for pushing up water into small heights in the stem, it can be responsible. How can we see that root pressure exists? On a day, choose a small soft-stemmed plant, and when there is plenty of atmospheric moisture, with a sharp blade early in the morning, cut the stem horizontally near the base. It is observed that the drops of solution ooze out of the cut stem, and this is because of the positive root pressure. To the cut stem, if you fix a rubber tube as a sleeve, actually you can collect the and measure the rate of exudation. Composition of the exudates also can be determined. When evaporation is low, effects of root pressure is also observable at night and early morning, and in the form of droplets. Excess water collects around special openings of veins near the tip of grass blades and leaves of many herbaceous plants. In its liquid phase, such a loss of water is known as guttation. In the overall process of water transport, root pressure can at best only provide a modest push. In water movement up tall trees, obviously they do not play a major role. To re-establish the continuous chains of water, molecules in the xylem may be the greatest contribution of root pressure, which often break under the enormous tensions created by transpiration. Most plants meet their need by transpiratory pull up to 15 meters per hour, as root pressure does not account for the majority of water transport. How is this movement accomplished? Through the plant, a long standing question is whether water is pushed or pulled. Through the plant, most researchers agree that water is mainly pulled and that the driving force for this process from the leaves is transpiration. This is called as the cohesion tension transpiration pull model of water transport. But what generates this transpirational pull? In plants, water is transient. For photosynthesis, the plant growth less than 1% of the water reaching the leaves is used. In the leaves, most of it is lost through the stomata. This water loss is known as transpiration. In an earlier class, you have studied transpiration by taking a healthy plant and enclosing it in a polythene bag and observing the droplets of water formed inside the bag. Using a cobalt chloride paper, you can also study water loss from a leaf which turns color on absorbing water. Transpiration. The evaporative loss of water by plants is transpiration. In the leaves, it occurs mainly through the stomata. In transpiration, besides the loss of water vapor, also in the leaf through pores called stomata, exchange of oxygen and carbon dioxide takes place. Normally, in the daytime, stomata are open and during the night they are closed. A change in the turgidity of the guard cells is the immediate cause of the opening or closing of the stomata. Towards the pore or stomatal aperture, the inner wall of each guard cell is thick and elastic. Within the two guard cells, when turgidity increases, flanking each stomatal aperture or pore, the thin outer wall bulge out and the inner wall are forced into the crescent shape. In the cell walls of the guard cells, due to the orientation of the microfibrils, the opening of the stomata is also aided. Rather than longitudinally, cellulose microfibrils are oriented radially, making it easier for the stoma to open. Due to the loss of water, when the guard cells lose turgor, their original shape is regained by the elastic inner walls. The guard cells become flaccid and the stoma closes. The lower surface of a dorsiventral leaf, often a dicotyledonous leaf, usually has a greater number of stomata, while they are about equal on both surfaces in an isobilateral leaf, which is often a monocotyledonous leaf. Several external factors are affected by transpiration: wind, speed, light, humidity, temperature. Transpiration affected by plant factors include distribution and number of stomata that are open, percent, the plant's water, status, structure of the canopy, etc. The ascent of xylem sap driven by transpiration depends mainly on the following physical properties of water. Cohesion, mutual attraction that is present between water molecules. Adhesion, to the polar surfaces, attraction of water molecules such as the surface of trickery elements. Surface tension, attraction of water molecules to each other in the liquid phase more than to water in the gas phase. A high tensile strength, that is, an ability to resist a pulling force, is given to the water by these properties, and high capillarity, that is, the ability to rise in thin tubes. Capillarity in plants is aided by the small diameter of the trachary elements, the vessel elements, and tracheids. Water is required for the process of photosynthesis. From the root to the leaf vein, the system of xylem vessel can supply the needed water. But to move water molecules into the leaf parenchyma cells where they needed, what force does a plant use? Through the stomata as water evaporates, since over the cell the thin film of water is continuous, it results in pulling of water molecule by molecule from the xylem into the leaf. Also, because in the atmosphere lower concentration of water vapor as compared to the substomatal cavity and intercellular spaces, water diffuses into the surrounding air. This creates a pull. Measurements reveal that by transpiration, the forces generated can create sufficient pressures to lift a xylem-sized column of water over 130 meters high. Transpiration and photosynthesis, a compromise. Transpiration has more than one purpose. It creates transpiration pull for transport in plants and absorption, supplies water for photosynthesis, transports minerals to all parts of the plants from the soil, cools leaf surfaces sometimes 10 to 15 degrees by evaporative cooling, maintains the shape and structure of the plants by keeping cells turgid. A plant that is actively photosynthesizing has an insatiable need for water. By the available water, which can be swiftly depleted by transpiration, photosynthesis is limited. The humidity of rainforests is largely because of this vast cycling of water from root to leaf to atmosphere and back to the soil. One of the strategies probably for maximizing the availability of carbon dioxide while minimizing water loss is the evolution of the C4 photosynthetic system. In terms of fixing carbon, C4 plants are twice as efficient as C3 plants. However, for the same amount of carbon dioxide fixed, a C4 plant loses only half as much water as a C3 plant. Uptake and transport of mineral nutrients. In the atmosphere, plants obtain their carbon and most of their oxygen from carbon dioxide. Their remaining nutritional requirements, however, are obtained from minerals and water for hydrogen in the soil. Uptake of mineral ions. All minerals, unlike water, cannot be passively absorbed by the roots. Two factors account for this. 1. In the soil, minerals are present as charged particles which cannot move across cell membranes, and 2. In the soil, the concentration of minerals is usually lower than the concentration of minerals in the root. Therefore, into the cytoplasm of epidermal cells, most minerals must enter the root by active absorption. In the form of ATP, this needs energy. For the water potential gradient in roots, the active uptake of ions is partly responsible and therefore for the uptake of water by osmosis. Some ions passively also move into the epidermal cells by both passive and active transport. Ions are absorbed from the soil. In the membranes of root hairs, cell-specific proteins actively pump ions into the cytoplasms of the epidermal cells from the soil. The endodermal cells, like all cells, have many transport proteins embedded in their plasma membrane. They let some of the solutes across the membrane but not others. The control points are the transport proteins of endodermal cells where a plant adjusts types of solutes and the quantity that reach the xylem. Note that because of the layer of suberin, the root endodermis has the ability to actively transport ions in one direction only. Translocation of mineral ions. Through active or passive uptake after the ions have reached xylem or a combination of the two, their further transport to all parts of the plant up the stem is through the transpiration stream. For the mineral elements, the chief sinks are the growing regions of the plant such as the lateral and the apical meristem, young leaves, developing flowers, seeds and fruits, and the storage organs. At the fine vein endings, unloading of mineral ions occurs through diffusion and active uptake by these cells. Mineral ions, particularly from older senescing parts, are remobilized frequently. Much of their mineral content are exported to younger leaves by the older dying leaves. In deciduous plants, similarly, before leaf fall, minerals are removed to other parts. Most readily mobilized elements are nitrogen, sulfur, potassium, phosphorus. Some elements that are structural components are not remobilized like calcium. Xylem exudates analysis shows that some of the nitrogen travels as inorganic ions. Much of it is carried in the organic form as amino acids and related compounds. Similarly, as organic compounds, small amounts of phosphorus and sulfur are carried. In addition, between xylem and phloem, small amount of exchange of materials does take place. Hence, it is not that we can clearly make a distinction and say categorically that phloem transports only organic materials while xylem transports only inorganic nutrients as was traditionally believed. Phloem transport flow from source to sink. By the vascular tissue, phloem food, primarily sucrose, is transported from a source to a sink. The source usually is understood to be that part of the plant which synthesizes the food, that is, the leaf, and sink is the part that needs or stores the food. But depending on the season or the plant's needs, the source and sink may be reversed. In the early spring, sugar stored in roots may be mobilized to become a source of food when the buds of trees act as sink. The energy is required for growth and development of the photosynthetic apparatus. Since the phloem, the direction of movement can be upwards or downwards, that is, bidirectional, the source-sink relationship is variable. The movement is always unidirectional in xylem, which contrasts with that of phloem, that is, upwards. Hence, in transpiration, unlike one-way flow of water, food in phloem sap can be transported in any required direction so long as there is a source of sugar and a sink able to remove, store, or use the sugar. Mainly water and sucrose is present in phloem sap, but through phloem other sugars, hormones and amino acids are also transported or translocated. The pressure flow or mass flow hypothesis. The accepted mechanism used from source to sink for the translocation of sugars is called the pressure flow hypothesis. At the source, by photosynthesis, as glucose is prepared, it is converted to sucrose, which is a disaccharide. In the form of sucrose, the sugar is then moved by active transport into the companion cells and then into the living phloem sieve tube cells. In the phloem, this process of loading at the source produces a hypertonic condition. Water in the adjacent xylem moves by osmosis into the phloem. The phloem sap will move to areas of lower pressure as osmotic pressure builds up. Osmotic pressure must be reduced at the sink. To move the sucrose out of the phloem sap and into the cells again, active transport is necessary, which will use the sugar converting it into energy, starch or cellulose. The osmotic pressure decreases and water moves out of the phloem as sugars are removed. To summarize, in the phloem, the movement of sugars begins at the source where into a sieve tube sugars are loaded. Loading of the phloem sets up a water potential gradient that facilitates in the phloem a mass movement. The sieve tube cells are present in phloem tissue, which form long columns with holes in their end walls called sieve plates. In the sieve plates, cytoplasmic strands pass through the holes, so forming continuous filaments. In the phloem, sieve tube is a hydrostatic pressure increases, pressure flow begins and the sap moves through the phloem. Meanwhile, incoming sugars at the sink are actively transported out of the phloem, and as complex carbohydrates they are removed. The loss of solute in the phloem produces a high water potential and water passes out, returning to xylem eventually. To identify the tissues through which food is transported, a simple experiment called girdling was used. A ring of bark up to the depth of the phloem layer on the trunk of a tree can be carefully removed. The portion of the bark above the ring on the stem becomes swollen in the absence of downward movement of food after a few weeks. This simple experiment shows that the tissue responsible for translocation of food is phloem and that transport takes place in one direction, that is towards the roots. Please share this video with your friends. Leave your comments below and please subscribe to my channel for more content. Click that bell notification that would let you know when a new video is uploaded. Thank you so much for watching. Once again, please subscribe to my channel for more content. For more videos, please check the description box down below.